Okay, so today we finish the urinary system. So urinary system part 2. Where are okay, let's finish this part first, then we'll go back. So you know that there are three processes of urine formation. One is called filtration, glomerular filtration. Another is tubular reabsorption, and the other one is tubular secretion. So filtration, reabsorption, and secretion, those are three processes of urine formation. Uh, for filtration, you need the pressure. The pressure that pushes the fluid out from the glomerulus is called the glomerular filtration pressure. And we'll see uh, what pressure causes the filtration in the glomerulus. And we'll talk about the renin-angiotensin system. What is this? Renin-angiotensin system is the part inside the urinary system or inside the kidney where the renin is produced and released. Renin is the chemical that regulates the blood pressure. We'll talk about the countercurrent mechanism of the formation of urine. What is that and how it works? Okay, countercurrent mechanism. We'll see the process of formation of dilute and concentrated urine. You know that the urine our body produces sometimes concentrated, sometimes diluted. So hormones control the status of the urine. If it is diluted or concentrated, that is controlled by the hormones. So we'll see which hormones and where it works. Then we'll talk about the urinary bladder and urethra. Okay, so those are the things we'll talk about in this part. First, these are three processes of urine formation: glomerular filtration, tubular reabsorption, tubular secretion. The pressure that causes the glomerular filtration is called the glomerular net filtration pressure, NFP, and that pressure is normally about 10 millimeter of mercury. That is the pressure that pushes the fluid out from the glomerulus into the nephron. Now, in that area where the glomerular filtration occurs, three pressures are present. One pressure is trying to cause the filtration, that means pushing the fluid out from the glomerulus, and two pressures oppose, act against it. Now, the pressure that is trying to push the fluid out or cause the glomerular filtration must be higher than other two pressures that are trying to stop it. That will cause the filtration, right? So we'll see what are those three pressures. You see here the blue arrows showing the pressure that is trying to cause the filtration, push the fluid out from the glomerulus. That is called glomerular hydrostatic pressure, and this pressure is about 55 millimeter of mercury, and two pressures are trying to stop that. One is the colloid osmotic pressure, which is 30 millimeter of mercury, and another one is the capsular hydrostatic pressure, which is 15 millimeter of mercury. These two are trying to stop the filtration. Now, you see here, if you add the two opposing pressures together, 30 and 15, that is 45, right, millimeter of mercury. And that pressure is trying to stop the filtration. And the pressure that is trying to cause the filtration is 55 millimeter of mercury. So the difference between 55 and 45 is 10 millimeter of mercury, right? And that is the net filtration pressure that causes the filtration. Now, how much filtration occurs in two kidneys in each minute? That is called the glomerular filtration rate. How much filtration occurs in two kidneys in 1 minute, and that volume is about 120 to 125 milliliter. Okay, so your both kidneys together cause that much filtration. Now, three factors are directly related to the glomerular filtration. What are those three factors? Total surface area for filtration, filtration membrane permeability, and net filtration pressure. Now, you see, if this is the glomerulus, this is the nephron here, right? You know that. Now, glomerular filtration moves the fluid from the blood into the nephron. That's the first step of urine formation, filtration. Now, the pressure this way is 55 millimeter of mercury. That is trying to cause the filtration. And opposing pressures, one is 30, another is 50. One is 30 millimeter of mercury and another is 15 millimeter of mercury. So together is 45, right? 45 millimeter of mercury. That is trying to stop, and 55 is trying to cause the filtration. So the difference is the net filtration pressure, which is what? 10 millimeter of mercury. You know that now. Now, if the pressure difference is more, for example, this one increases to 65, then the difference will be 20, right? The difference will be 20. So more fluid will enter here. Filtration will be more. So pressure difference more means the net filtration pressure is more, right? Higher. So that will cause more filtration. That is clearly understandable. Now another thing is total surface area. What is that? Now you see this way. This is the glomerulus, ball of capillary, right? It's very long, you know that, because it is a ball. Now, if this is the glomerulus of one person and another person's glomerulus is much smaller, for example. So which one has more surface area for filtration? This one. Okay. If you make it straight, this one has more surface area. So how much surface area is available? That is another factor, right? More surface area, more filtration. Because blood will stay there for a longer time. Another is permeability. Now is think that two persons both have same sized glomerulus, same sized surface area, okay? But one person's glomerulus is healthy, so it has more openings. In this one, you have many openings closed by infection or something, okay? Make sense? So many pores got closed or blocked. So although the surface area is same, but the permeability more in this one, right? So the fluid will pass more easily through this one. So those are the three factors that can directly influence the filtration, how much filtration will occur. Now, you now know how filtration occurs here. After the filtration, fluid is now here. Fluid enters into the first part of the nephron and then it starts to move. At different locations, reabsorption, that means taken back into the body. Always remember, reabsorption means taken back into the body, going back into the body. And secretion occurs. Secretion is just opposite of reabsorption. That means entering into the nephron, getting out from the body. Okay, so this is secretion and this is reabsorption. Okay. So in the tubule, reabsorption as well as secretion occurs. Okay. So now you just think and tell me, if your body wants to excrete anything, it will cause secretion or reabsorption. Secretion. Make sense? If wants to preserve anything or retain anything, reabsorption. Is it clear? Okay. Okay. Now, when the fluid passes through the proximal convoluted tubule, this part is called the proximal convoluted tubule. Okay. 65% of everything is reabsorbed here equally. That means water and other chemicals are equally taken back into the body. Is it clear? Okay. So, if everything is reabsorbed equally, the concentration will remain same or change. Same, right? Like if you get a glucose sugar solution, if you take the water and sugar equally out, the volume will go down, right? But the concentration will remain same. So here, the volume will go down 65%. That means if it is 100 here milliliter, here it will be, when it will come here, the volume will drop down to 35 milliliter because 65% of it will be reabsorbed. Make sense? Okay. But the concentration is same here and here because everything will be equally reabsorbed. Okay, only the volume will significantly go down. Now, this is the loop of Henle. When the fluid will move down to the descending limb of loop of Henle, what will happen here? In this part, only water will be reabsorbed because this wall only allows the water to pass. So only water will be reabsorbed. Now you tell me, if you have the solution, again sugar solution, if you only take the water out, then the concentration will go up or down? Up, right? Because you are taking the water part out. So sugar is still there. So the concentration will go up, but the volume will go down, right? So same thing will happen here. You are taking only the water out, but other electrolytes or chemicals are inside, right? So the concentration will start to go up, up, up. And when it will reach here, the concentration will be highest, right? The concentration will be highest in the bottom of the loop of Henle. Is it clear? Okay. Here, if you measure the concentration, you see the concentration is from 100 to 300 milliosmol. That's the unit of concentration or osmolarity. But when it goes down all the way to the bottom of long loop of Henle, that means juxtamedullary nephron, long loop. Here, it reaches to 1200 to 1400 milliosmol. So very high concentration here. Okay, because a lot of water is reabsorbed through the wall of descending limb of leukopen. Now, when the fluid will start to move up through the ascending limb of loop of Henle, what will happen here? This one is only permeable. The wall is only permeable to sodium, chlorine, potassium. it will not let the water get out or reabsorb. So this one is only for water, this one is not for water but other chemicals. Now you tell me, if the fluid goes up and you take the solutes out, water is remaining there. It's like, in the, again, sugar solution, you are now taking the sugar out, right? Leaving the water there. So the concentration will go down because water ratio will go up. So the concentration is going down, down, down. When it reaches here, it will begin like this. But what will happen when the fluid will come from here to here? A lot of fluid is already taken back. The volume will significantly go down, right? Make sense? Because everywhere actually the volume is going down, but the concentration is going in opposite direction. Now, when up to here, that means in the first part of the nephron, proximal convoluted tubule and loop of Henle, no hormone works. No hormone works here. Hormone works on the last part of the nephron. That means what? The hormones work on the distal convoluted tubule. This is the distal convoluted tubule here, and on the collecting duct. In these two structures or parts, the hormones work. These two are sensitive, highly sensitive to hormones. Now, let's see what will happen here. In the distal convoluted tubule, which hormone works? Aldosterone. So, in the distal convoluted tubule, mainly aldosterone hormone works. Aldosterone comes from the adrenal cortex. You know that adrenal gland sits on the kidneys and it has outer cortex and inner medulla. You must know this from NP1. Aldosterone is an adrenal cortical hormone. That means comes from the adrenal cortex and works on the distal convoluted tubule of the nephron. Make sense? Okay. Selective action. And what it does on the distal convoluted tubule? What it does is it reabsorbs sodium and water here. So aldosterone reabsorbs sodium and water from the distal convoluted tubule. Sodium and water. Make sense? So if more aldosterone is released, then what will happen? More sodium and water will be reabsorbed. Is it clear? That means they will enter back into the body more. Go back into. So the volume of blood will go up or down? Up, because water is going back, right? Sodium is going back. The volume will go up. So aldosterone will increase the blood volume and increase the blood pressure. Make sense? More blood volume means more blood pressure. On the collecting duct, the hormone that works is called antidiuretic hormone or ADH. Antidiuretic hormone or ADH. It comes from the posterior pituitary. That means the posterior lobe of the pituitary gland in your brain. So you see, interestingly, it is produced in the pituitary gland, right? And goes all the way down to the kidneys. Not only kidneys, it only works on the last part of the nephrons, not everywhere. So ADH reabsorbs water in the collecting duct. So antidiuretic hormone reabsorbs water. Its name tells you, right? Its function. What is? Have you heard the word diuretics? Diuretics produce more or less urine? Diuretics. More. More, right? Diuretics produce more urine. So antidiuretics will do what? Less, right? Antidiuretic. diuretics produce more, antidiuretics will produce less. So antidiuretic hormone produces less urine. That how? To produce less urine, you have to take the water back, right? Reabsorb the water. So that's how antidiuretic hormone works. It works on the collecting duct mainly and reabsorbs a lot of water. That means takes a lot of water back into the body. So the volume of urine will be less. Make sense? So more ADH means less urine. Does it make sense? Less urine. Now there is a clinical condition that is called diabetes insipidus. What happens in diabetes insipidus usually happens due to the destruction of posterior pituitary due to tumor or cancer. And destruction of posterior pituitary, when that happens, posterior pituitary cannot produce ADH, right? Because of destruction of posterior pituitary, the amount of ADH goes down. So if less ADH is produced, that means what? More urine will be formed, right? Because ADH takes the water back into the body. But if you don't have ADH, then what will happen? Water will not go back into the body. So water will get out from the body as urine. Is it clear? So diabetes insipidus is the clinical condition where the patient produces excessive amount of urine, too much urine production due to decreased release of antidiuretic hormone. And why that happens? Usually due to tumor or cancer of the posterior pituitary gland, destruction of the posterior pituitary gland. Okay, so now you tell me, antidiuretic hormone reabsorbs water, decreases the volume of urine, right? That means increases the volume of what? Blood. Make sense? Water is taken back, volume of urine goes down, but the volume of blood will go up. So it will increase the blood pressure. Make sense? More antidiuretic hormone, it will increase the blood pressure. You know that when the patient is taken to the emergency urgent care with high blood pressure, hypertension, right? Usually they give Lasix, the medicine that will produce more urine. That will decrease the volume of blood, right? Will decrease the blood pressure quickly. So what that medicine does, blocks the ADH action on the tubule, collecting that. Make sense? So if you block the ADH action here, then ADH will not be able to reabsorb water. Water will get out from the body quickly. Make sense? So that's how many medicines, blood pressure medicines work. Okay, so just remember what I have mentioned here. Secretion. Which chemicals should be secreted in the tubule? You already know. Those chemicals we don't need in our body, right? Secretion will excrete out from the body. So, which chemicals are secreted? Urea, uric acid, right? Uh, those are the harmful chemicals produced in our body. Those are mainly secreted from the body. If the hydrogen ion concentration increases in your body, hydrogen ion concentration increases, that will cause acidosis, right? More hydrogen, less pH, more acidic, right? So in that case, hydrogen ion will be secreted. Plenty of hydrogen ion will be secreted to bring the pH back to normal. Renin angiotensin. Renin angiotensin system that controls the blood pressure. How? Inside the kidney, although when we see the nephron in the book, in the picture, or when I draw, I expand the nephron a lot to show you different parts. But inside the kidney, just think that you have 1 million nephrons in one kidney, right? So there is not enough space for their neurons to spread. Everything is highly squeezed, right? Heavily squeezed. So inside the nephron, what happens actually inside the kidney? This is the nephron, okay, uh, very much squeezed like this. Uh, Let me draw again. So uh, okay, so see here, this is the proximal convoluted tubule loop of Henle, right? And distal convoluted tubule is here. And this is the afferent arterial glomerulus efferent artery. Okay. So what happens inside the kidney, the distal convoluted tubule and the afferent arterial, these two structures are side by side. And these two structures together called the juxtaglomerular apparatus, JGA, juxtaglomerular apparatus. Okay. Afferent arterial and the distal convoluted tubule. And now, if you see the wall of afferent arterial here, in the wall of afferent arterial, you have highly specialized cells only in this part, in juxtaglomerular apparatus part. And these cells are called the granular cells. Granular cells. And these granular cells releases renin, the chemical. Okay? So the granular cells of afferent wall of the afferent arterial produces renin. And when the blood pressure inside the arterial will go down, decreased blood pressure will stimulate these granular cells to release renin. So decreased blood pressure, when the blood pressure goes down, that stimulates the granular cells to release the renin. Okay. Usually, if the mean arterial pressure, you know that diastolic plus 1/3 of pulse, right? Mean arterial pressure goes below 80 millimeter of mercury. That causes the release of renin from the granular cells of juxtaglomerular apparatus. Now, how the renin works? Renin is an inactive chemical. It cannot work directly. It is because it is inactive. Renin converts the angiotensinogen which is present in the blood. Angiotensinogen. Renin works on it and converts the angiotensinogen to angiotensin 1. Angiotensin 1. Okay. And then uh, an enzyme that is called angiotensin converting enzyme, ACE, angiotensin converting enzyme, that converts the angiotensin 1 to angiotensin 2. This one is present in the liver and lung. Okay. So angiotensin converting enzyme that converts the angiotensin 1 to angiotensin 2. Now the angiotensin 2 causes generalized vasoconstriction. Vasoconstriction. You know that. Generalized means everywhere in the body, right? Not localized. Generalized vasoconstriction. Constriction. Constriction. Vasoconstriction will increase or decrease the blood pressure. Increase the blood pressure, right? Vasoconstriction everywhere in the body occurs, and that causes the increase of blood pressure. Make sense? So the blood pressure will come back to normal. So renin is released when the blood pressure goes down, becomes low, and that produce the angiotensin 2 that causes generalized vasoconstriction to increase the blood pressure. That's the main action of angiotensin 2, generalized vasoconstriction. Angiotensin 2 has other actions too. Stimulates the reabsorption of sodium on the tubules. So more sodium will be reabsorbed, and when the sodium is reabsorbed, remember sodium has a tendency, sodium is reabsorbed, sodium always tries to hold water, okay, with it. So when sodium will be reabsorbed, some water will be also reabsorbed. So the blood volume will increase, that will increase the blood pressure. Angiotensin 2 also stimulates the adrenal cortex to release aldosterone. That what I have talked about that. That also causes the reabsorption of sodium and water from the distal convoluted tubule. We have just talked about that. So more aldosterone will be released from the adrenal cortex that will reabsorb sodium more and water. Okay, increase the blood volume, increase the blood pressure. So you see in different ways, angiotensin 2 increases the blood pressure. The strongest one is generalized vasoconstriction everywhere. Stimulates the hypothalamus to release more ADH, and ADH will do what? Reabsorbs water in the collecting duct. We have talked about that, right? Comes from the posterior pituitary and will reabsorb more water. So basically, angiotensin 2 causes generalized vasoconstriction and increase the blood volume by reabsorbing more water. So that's the main, those two are the main actions. Another thing interesting you see here, also activates the thirst center, angiotensin 2. That means what? When your thirst center will be activated in the hypothalamus in the brain, you will feel what? Thirsty. Because thirst center in the brain is activated. So if you feel thirsty, what you will do? Drink more water. So blood volume will go up, blood pressure will go up. So that is another way it will increase the blood pressure. So it will increase the blood pressure in different ways. Generalized vasoconstriction, release of aldosterone, release of ADH, right? And stimulating the thirst center. Now, the counter current mechanism of the formation of urine. So, again, let me draw the nephron, proximal convoluted tubule, loop of Henle, distal convoluted tubule, collecting duct. You know this part. And this is the loop of Henle. It has descending limb, ascending limb, two limbs. Now, you remember in long looped nephrons, you have what? Vasa recta. In long looped nephron, you already know there are three sets of capillaries. We have talked about that. So this is the glomerulus here. This is the efferent arteriole. And what happens in case of long looped nephron, the efferent arteriole goes like this. Okay, along the long loop of Henle, and it is like ladder. this capillary structure is like a ladder, and this is called what? Vasa recta. Do you remember that? Okay. So now we already know that when the fluid goes down to the descending limb of loop of Henle, who is reabsorbed here? Water. Very good. A lot of water comes out. Water comes out from the neck. And fluid is going this way, and here the fluid is going this way, right? And when the fluid goes this way, sodium, chlorine, potassium, those are reabsorbed, right? Okay. Now the blood that flows, actually it goes actually this way. Here, this way, and then that's all this way. So when the blood enters into vasa recta, flows in this direction. So you see here, let's see here, the fluid is going this way, right? And blood is going this way, side by side, in opposite direction, right? Opposite direction. That means counter current. The flow is counter means opposite. Also here, in opposite direction, right? So, and they are side by side. The limb of vasa recta and limb of loop of Henle, they are side by side. Now, why this type of arrangement is essential? You see here, when water leaves the descending limb of loop of Henle, that water will enter into the capillary here. Makes sense? Quickly will enter into the blood. When sodium, chlorine, potassium will leave, they will quickly enter into the blood here. Okay. Now you tell me, when water leaves here, the concentration goes up or down? Up, right? Concentration, because water is leaving, right? But when water enters into the capillary blood, the blood concentration will go down because water is getting in, right? Water is leaving here, but water is getting in quickly, right? Makes sense? What happens here? When the sodium, potassium, chlorine, they get out, concentration goes up. Concentration goes down, right? Because they are leaving, water is remaining. We have talked about that. Concentration is going down. But they are quickly entering into the blood, right? So blood concentration is going up. Is it clear? So not only the direction of flow is opposite, but also the change of concentration is opposite, right? Because water is leaving, but also entering into the blood at the same time. So that's why we called this as counter current mechanism of urine formation, which is essential because you just now think, if you didn't have the vasa recta here, okay, just think that you don't have vasa recta here, what will happen? Water will get out, then a lot of water will accumulate here, right? Because there is no capillary to remove it. So what will happen? Water will start to accumulate here, right? The pressure of water will increase here. So no more water will be able to get out. So urine production will not occur, right? So that's why the vasa recta is continuously removing the water and sodium, potassium, chlorine that are being reabsorbed. Make sense? Someone has to clean that area, just take them away, right? So new reabsorption will occur. Otherwise, the things will accumulate there, will not let further reabsorption to occur. So the countercurrent mechanism and the presence of vasa recta is very important, right? There. Otherwise, the urine production will not occur. Just know that much. Here, if you see, take a small amount of fluid from the outer part of the cortex of the kidney and from the very inner part of the medulla. You will see the concentration difference is high. The outer part is about 300 and in the innermost part about 1200, right? That actually I have explained to you. You remember here the concentration I said what? milliosmol. Remember that? And here I said 1200 to 1400, right? So in your kidney, the cortex contains this part of nephron, and the medulla contains this part of nephron, right? If you go in the deeper part of medulla, you'll get this, right? So high concentration in deeper part of medulla, but in the cortex, you'll see the concentration is less there. Although you have millions, but that's how the nephron is located. The cortex, these parts, and in the medulla, this part. Now you see in the picture that yellow nephron. Nephron here, right? So the nephron look friendly. I have talked about that. This is the vas rector. Urinary bladder. Urinary bladder holds the urine for few to several hours, which is very important because kidneys continuously produce urine, right? So someone has to hold the urine until you urinate. That is done by the bladder. Bladder has three layers. If you see the wall of the urinary bladder, innermost layer is the epithelial layer, which is formed by transitional epithelium. Middle layer is thick layer, muscular layer, which is formed by a special type of smooth muscle, which is called detrusor muscle. So middle layer is thick and smooth muscle layer, and that smooth muscle is called detrusor muscle. And outermost layer is the adventitia, fibers adventitia. That one mainly protects protection. Now, why the innermost epithelial lining is transitional? Anybody? Yeah, expand. You remember I said that transitional epithelial cells can get flat and elongated, right? Change the shape. So that is very important because your urinary bladder expands and contracts, right? So the epithelial lining can expand and contract because the cells can change their shape. That is number one. Number two, transitional epithelium doesn't allow any fluid to pass through it. Doesn't allow any what? Fluid to pass through it. Which is very important here because urinary bladder does what? Holds the urine, right? If the fluid or urine passes through the epithelial lining and enters into the body back, no point of producing urine, right? If it can go back. It must not go back into the body. So that's why you have transitional lining that will not let the urine get back into the body, pass through. Okay, so those are the three layers of the urinary bladder. Now, here you see the upper part of the bladder has an additional outer membrane or layer. What is that? Just think that this is your abdominal cavity and this is the pelvic cavity. This is your urinary bladder and this is the body wall. You know that parietal peritoneum. Do you remember? Peritoneum is attached to the body wall, inner surface of the body wall. And you know the mesentery visceral peritoneum. We have talked about that in digestive system, right? So parietal peritoneum goes down like this, and like this. So now this is the wall of the bladder. So here you don't have that peritoneum, but if you see the upper part of the urinary bladder, upper surface of the urinary bladder, you have the peritoneum covering. Make sense? So that's why you have an additional peritoneal layer in the outer part of the bladder, uh, only the upper part, that is the peritoneum. Now, inside the urinary bladder, you have three openings. You know that. Ureters bring the urine from right and left kidneys, right? So, two ureters enter into the bladder, and also you have the opening for the urethra that takes the urine out from the bladder. Now, if you join those three openings by imaginary line, there is no actual line. Just think that lines are joining those three openings inside the bladder. These two are for the ureters, and this is for the urethra. And this part only is very smooth. The surface is very smooth and shiny. This is called the trigon. Okay, this part is. Rest part is rough. A lot of foldings are the rest part of the bladder, inside the bladder, and those foldings are called rugae, same as the stomach, right? Inside the stomach you have the folds called gastric rugae. In the bladder you also have rugae. Only the trigon is smooth and shiny, no foldings there. Trigon acts as a funnel. You know funnel? You pour the liquid, right, into small thing through the funnel. So trigon is like this, works as a funnel and pours the urine into the urethra. Okay, helps to pour the urine into the urethra. So if that area is rough, trigon is rough, what will happen when the urine will enter into the urethra? Some urine will stay there, right? Will not easily, smoothly enter into the urethra. So that's not good. So that's the bladder. Urethra. Uh, this is the part of the urinary system which is different in male and female. Female urethra is very short, male urethra is very long. Uh, Female urethra is 3 to 4 centimeter long and male urethra is 15 to 20 centimeter long. Okay, 15 to 20 centimeter long. Now, in male urethra, you have three parts: prostatic part, membranous part, and spongy part, or prostatic urethra, membranous urethra, and spongy urethra. Those are three parts. First part is inside the prostate, that is called the prostatic urethra. Here is the uh, inside the prostate. This is the first part, which is the prostatic urethra, and second part is very short, 2 centimeter, and that is the membranous urethra. Why this is called membranous? Because this part is located surrounded by a diaphragm that is called urogenital diaphragm. This one. And inside the urogenital diaphragm, that part is the membranous urethra. And the last part is very long. It's about 15 centimeter long. That is inside the penis. That's the spongy or penile urethra. Now, the urethra has three different types of epithelial lining that you have to remember. Three different types. Actually, that makes sense if you think carefully. The first part of the urethra, this is the urinary bladder, and this is the first part of the urethra. What is the epithelial lining of the bladder? Epithelial lining of the bladder is transitional, right? Transitional. So what happens? That transitional epithelial lining enters into the first part of the urethra. Continuation of that, right? Because urethra starts from the bladder, right? So transitional from the bladder enters a little bit into the urethra. So the first part, very first part of urethra is transitional. Then The middle long part is ciliated columnar, pseudo-stratified ciliated columnar is the long middle part, and only the last part is stratified squamous. Very last part is what? Stratified squamous. Where you find the stratified squamous? I have mentioned 100 times. Where? Those areas you have friction, right? So the last part, the chance of getting friction is more, right? Or getting exposed to air is more. So the last part is stratified squamous. Makes sense. First part is transitional because it's the continuation of the bladder, right? From the bladder, little bit comes. So, but middle long part is pseudostratified ciliated columnar. Those are three types of lining. In the urethra, you have two sphincters. One is here, internal, and another is here, external. Now, this one is skeletal, like your anal sphincter, skeletal muscle. And this one is what? Smooth. So which one we can control? The external, right? External one, when we desire to urinate, we voluntarily open this one, relax this one, right? Now what should be done? This one should be open, right? So when we relax the external urethral sphincter, signal goes to the spinal cord, and from there signal comes back here, right? To open this one, because we cannot do that. So when both are open, the urine will get out. Let the urine get out. Okay, so that's the reflex of the urination is controlled. Okay, so now just I'll mention a couple of clinical conditions related to the urinary system. Probably you have already heard Antidiuretic. A means what? Absence, right? Apnea, no breathing. Anuria, no urine. Make sense? Anuria is no production of what? Urine. No production, or if the production of urine is very small, like about less than 25 milliliter per day, that means almost no urine production. In that case, we say anuria. No or very small amount of urine production. Oliguria, decreased volume of urine, decreased production of urine. Okay. Decreased production of urine. If it is less than 200 milliliter per day, we consider that as oliguria. Highly concentrated, decreased amount of urine. Polyuria. This is opposite, right? Poly means more. Polyuria is increased production of urine. So the volume of urine will be more than normal in polyuria. Hematuria. What is this? Blood in urine. Blood in urine. We see blood breakdown products, or sometimes we see whole red blood cell in the urine. Hematuria. Proteinuria. You can add more. Proteinuria. Proteins in urine, right? If you see the presence of protein in urine, that's called proteinuria. If you see specific type of protein, albumin, that is called what? Albuminuria. Albuminuria. So proteinuria in general, but if you get a specific one, we can term that way. So those are different conditions to diagnose the diseases in the urinary system.